Genomics is the study of whole sets of genes and their interactions. Key terms for genomics include metagenomics, in which DNA from a group of species is collected from an environmental sample and sequenced. Bioinformatics is the application of computational methods to the storage and analysis of biological data. Gene annotation refers to the identification of protein-coding genes in a sequence and their functions. Pseudogenes refer to former genes that have accumulated mutations over a long time and no longer produce functional proteins. Proteomics refer to the studying of full protein sets. Comparative genomics compare different genomes to assign gene functions. Orthologs are genes that occur in different species but have a clear sequence and functional relationship to each other. Gene characterized in one species can be used to tentatively assign gene function to the ortholog in another species. Paralogs are genes similarly related to each other within a single species. And synteny refers to a conserved order of genes of closely related species, which provides additional evidence for an orthologous relationship between genes at identical locations within the related segments. Evolutionary developmental biology, or Evo-Devo, is the study of the evolution of developmental processes in multicellular organisms. Genomic information shows that minor differences in gene sequence or regulation can result in major differences in form. Hox genes, also known as homeobox-containing genes, are highly conserved among different animals. Small changes in regulatory sequences within Hox gene may lead to major changes in body form. FOXP2 gene is required for the proper development of speech and language in humans. DNA sequencing is central to the study of genomics. There are three main methods of DNA sequencing. The Maxim-Gilbert sequencing is based on nucleobase-specific partial chemical modification of DNA and subsequent cleavage of DNA backbone at sites adjacent to the modified nucleotides. Sequence is determined by gel electrophoresis. Sanger sequencing is based on dideoxynucleotides, which are modified nucleotides that would result in chain termination. Fluorescently labeled dideoxynucleotides are added to target DNA, producing fragments of different lengths. Sequence is also determined by gel electrophoresis. Next-generation sequencing is a modern DNA sequencing technique that is a lot quicker and cheaper than Sanger sequencing. It is essentially running a very large number of tiny Sanger sequencing reactions in parallel. The sequencing of the human genome, known as the Human Genome Project, began in 1990. The project involves 3 stages. The linkage mapping stage maps the location of several thousand genetic markers on each chromosome. A genetic marker is a gene or other identifiable DNA sequence, including single nucleotide polymorphism, abbreviated as SNPs, which differ by a single nucleotide, and short tandem repeats, which differ by the number of repeats. The physical mapping stage involves the ordering of large overlapping fragments, such as cloning factors, including yeast artificial chromosome, abbreviated as YAC, and bacterial artificial chromosome, abbreviated as BAC, as well as smaller fragments such as plasmids. Finally, DNA sequencing is used to determine the complete nucleotide sequence of each chromosome. A complete haploid set of human chromosomes consists of 3.2 billion base pairs. The human genome only consists of 27.5% of genes, including 1.5% of exons, which codes for functional proteins, and 26% of introns, which may be spliced out during RNA splicing. 44% of human genome consists of transposable elements, which can move from one site to another in a cell's DNA, which includes 10% of ALU elements that are found in humans and primates, 20% of lines, which stands for long interspersed nuclear elements, and 13% of sines, which stands for short interspersed nuclear element. Lines and sines are retrotransposons that amplify themselves through RNA intermediates. 16% of the human genome consists of repetitive sequences, including 3% of short tandem repeats, also known as microsatellites, which are repetitive DNA less than 10 base pair long that vary in the number of repeats among different individuals. As I mentioned in my video on biotechnology, short tandem repeats are very useful in genetic profiling for forensics or paternal testing. 5% of human DNA are segment duplications, and 8% are long repetitive sequences, such as telomeres and centromeres. The rest of the human genome are miscellaneous elements. The genomes of most bacteria and Archaea range from 1 to 6 million base pairs, abbreviated as MB. Genomes of Eukaryotes typically range from 10 to 4,000 million base pairs. Bacteria and Archaea typically contains 1,500 to 7,500 genes, whereas eukaryotes contain 5,000 to 40,000 genes. The number of genes is not correlated to genome size. Vertebrate genomes can produce more than one polypeptide per gene because of alternative splicing of RNA transcripts. In general, bacteria and Archaea have higher gene density than eukaryotes. Humans and other mammals have the lowest gene density, or numbers of genes in a given length of DNA. Multicellular eukaryotes have many introns within genes and non-coding DNA between genes. Whereas bacteria lack introns, Archaea only has some introns, and both of them have little non-coding DNA.